Hello, everyone. So I finally, I think that I might have my internet upgraded. We'll see. I totally changed companies, so we'll see how that goes. Um, anyway, I hope we can do the class synchronously on Thursday. I might even, if the internet isn't like changed over and up on Thursday, I may go up to campus. But on last Thursday on Teams, I discussed a number of things about vascular physiology. Now, we talked about cardiac physiology, which has to do with cardiac output, stroke volume, um, heart rate, the sympathetic nervous system increasing or actually decreasing the latency between heartbeats, so increasing heart rate that we measure in beats per minute. Hopefully, many of you, I haven't checked the sheet on the Google sheet, but hopefully I'll get that done in the next couple of days. It's like a 10-minute exercise. So please go ahead and do that, because as soon as we do that, people can start analyzing the data and making the graph, and you can just get that off your plate. I know it's a nice thing to get things off your plate. But so last Thursday, just to recap, we talked about flow, and I'm going to just kind of highlight here in a couple of different ways. Flow, mean arterial pressure, and really I say they're in opposition because as mean arterial pressure increases, that means the tension of the arteries is increasing, which is going to oppose flow. But as flow goes up, pressure is going to go up, and that's just because you're putting more fluid through the hose or the artery. But as arteries clamp down, that's going to decrease flow. So it's an antagonistic relationship, but understand in some ways, as flow goes up, mean arterial pressure is also going to go up. So sympathetic activity increases heart rate through the sinoatrial node, and that's through those pacemaker cells, increases stroke volume, and that's through increased contractility of the ventricles of the contractile cells, and by increasing venous return. And it does this by stimulating those smooth muscles via alpha 1 receptors to contract, constricting those. We talked about mean arterial pressure, just defining it as systolic pressure plus 2 times the diastolic pressure. And the reason we double diastolic pressure is because it lasts twice as long as the systolic phase. And divided by 3 because you have 1, 2, 3 units here, and we get a mean pressure or a mean arterial pressure. We moved from there into a number of just terminologies, different ligands right here and right here that will increase blood flow. Or actually, here they increase, carbon dioxide increases blood flow, whereas serotonin and vasopressin, it's right there in the names, toning up and pressing down. Serotonin and vasopressin are vasoconstrictors. Norepinephrine and epinephrine, sympathetic nervous system, those are also vasoconstrictors. One interesting thing is norepinephrine and epinephrine can act on blood vessels via beta receptors. Beta receptors relax smooth muscle. So that's going to increase blood flow, and you have beta receptors in specific places, say in your muscles. Because when your sympathetic nervous system is activated, you want more blood flowing to your semimembranous and semitendinous muscles and your biceps femoris muscle. Those muscles in your legs that allow you to run away from the baboon, right? So angiotensin, we talked about the RAS system, the renin system, aldosterone system, and angiotensin system that increase sodium and lead to water retention, which is going to increase blood pressure by increasing the volume of blood in your body. Finally, we talked about capillaries. And go back and listen to this. I listened to the recording, and I think I came through pretty clear where at the arterial end of capillaries, you have a hydrostatic pressure that surpasses the osmotic pressure. As you move through the capillary beds, which are really leaky, you lose hydrostatic pressure. It goes down to almost zero, down to 15 millimeters of mercury. But because the proteins in blood do not leak out, osmotic pressure stays the same, and you reabsorb all the blood. Here at the arterial end, you facilitate filtration, meaning you filter stuff out. Here at the venous end, you favor absorption. Now that is a great segue to start talking about blood. Blood physiology. This is really the physiology of hemoglobin, but we'll talk about a few other things. Your serum proteins, or you could call these plasma proteins also, are albumins, globulins, and fibrinogens. So these three proteins make up the osmotic pressure. They keep the osmotic pressure high in the blood. They maintain it at about 35 millimeters of mercury. These are the valuable things in blood, and this is why people buy blood plasma, for these proteins. Albumins include carrier proteins, so all of those things that allow steroids to flow in the blood, all of those things that protect protein hormones from degradation are albumins. Globulins are our antibodies. These are very important, especially now we're talking about antibody therapy as a potential therapy for treating people with intense sickness from coronavirus. If the disease that results from coronavirus, they call it COVID-19, if that gets very severe, individuals can be given antibodies from individuals who have been exposed to the virus recently. And then fibrinogens, these are clotting factors. So all of these are proteins in the blood that maintain osmotic pressure. The process of making blood is called hematopoiesis. And hematopoiesis is stimulated by the intima cells. These are the epithelial cells of the tunica intima. This layer of the blood vessel secretes paracrine signals that stimulate multiple processes. And these are erythropoiesis, formation of red blood cells. This is stimulated by a molecule called erythropoietin, named kind of logically. Thrombopoiesis, which is stimulated by thrombopoietin. Thrombopoiesis is the formation of megakaryocytes. And megakaryocytes, remember, split off chunks to form platelets. And then finally, a process that we can term leukopoiesis, but this is very diverse. Leukopoiesis is the process of forming white blood cells. And white blood cells are varied. You have granular and agranular, and they serve many different purposes in immunity, in recognizing out of control cells. They're kind of the cleanup, and then they serve many different functions in the body. So leukopoiesis is going to be the most diverse. So we have many different ligands that stimulate leukopoiesis. Colony stimulating factor is one. Interleukins, and there are multiple of these. A couple of you are writing papers that center on interleukin physiology. Stem cell factor, and many other factors that really just stimulate the process of white blood cell formation. So these processes that are involved in hematopoiesis occur in multiple places. Occurs in the spleen, liver, bones. The number of bones that hematopoiesis occurs in changes or decreases throughout our life until it's only left in the pelvis, ribs, some bones of our skull. So all of these have in common, they're all flat bones. And at the very ends of some long bones. And remember, those are like our femur, our humerus, radius and ulna, but there's not too much hematopoiesis that goes on in the smaller long bones. It's primarily the femurs and the humeruses. So red blood cells are replaced about every 6 months. I should say their lifespan is about 6 months. White blood cells are much shorter lived. They live a few hours. So there's a big difference there between red blood cells and white blood cells and the amount of time that they live. One of the things that's going to stimulate erythropoiesis, which remember is the formation of red blood cells, is a transcription factor called HIF1. And this is hypoxia inducing factor. Which makes sense. Red blood cells, the main thing that they do, really the only thing that they do, is carry oxygen. So when you're hypoxic, you don't have enough oxygen, hypoxia inducing factor. I'm sorry, that's actually hypoxia inducible factor. It doesn't induce hypoxia, it's induced by hypoxia. So hypoxia inducible factor 1 is a transcription factor, and this transcription factor increases the transcription of a protein called erythropoietin. So that's pretty cool, right? Now, the reason that red blood cells only carry oxygen is because they lose all organelles as they mature. There's no mitochondria. There's no nucleus even. Remember, a synapomorphy of mammals is an anucleic, or without a nucleus, red blood cell. They don't have an endoplasmic reticulum. They don't have any ribosomes. They really don't have anything at all. The only thing they have is a protein that they're packed with called hemoglobin, and they have tons and tons and tons of hemoglobin. So it's very important for blood to be as packed as possible with red blood cells, because the primary reason that we have blood is to carry oxygen. Now, things like the endocrine system have, and the kidneys have co-opted blood to clean toxins out of the blood, to send signals by the blood in the case of the endocrine system. But the primary reason we have a circulatory system is to carry oxygen from our lungs all around our body, because remember, we're so big that we can't diffuse oxygen to all of our cells. So we have blood to carry oxygen, and how blood carries oxygen is via red blood cells, and the only thing that red blood cells have in them is hemoglobin, which is a special protein that carries oxygen. So let's talk a little bit about how hemoglobin can carry oxygen. So this is actually a photo of a red blood cell. And it's probably a drawing more. But if you look at red blood cells under a microscope, this is actually what they look like. They're a bit concave, and this increases the surface area. But remember, inside of this cell membrane is only hemoglobin. And this is a 4 subunit protein. You have a alpha chain. Let's decrease the size of our little marker here. So you've got an alpha chain, a beta chain, another beta chain, and an alpha chain. And we'll talk about the significance of the different chains that make up the 4 subunits of hemoglobin in a moment. But the functional group, or the functional part of this protein, is this central heme group that is an iron atom bound to 4 nitrogens. And this iron atom has a high affinity for oxygen. And the reason it has a high affinity for oxygen is it loves to give away electrons. Just like we talked about back in the citric acid cycle, oxygen loves to take electrons, and iron can be oxidized like crazy. It'll reduce oxygen as much as it wants to be reduced. So oxygen comes in and binds to iron, and it takes it to wherever in the body it needs to go. Now, in healthy individuals, red blood cells make up about 50% of the blood, give or take a few percentage points. But in healthy individuals, we'll see that half of the blood is made up of these red blood cells. And what you can do is you can take a little tiny tube called a capillary tube. This is about, we'll say, 500 microns across. And you may have seen somebody do this. Somebody will take your finger and prick it with a little pin, and you secrete a little bit of blood, and they'll take this capillary tube and just touch it to the blood. And what happens is the blood is going to seep up the capillary tube, filling it, and then they centrifuge or spin around really quickly this capillary tube, and you will end up with something that looks like this. Oops. Hopefully not blue. About halfway up, it's really dark red. And the other half of the way up, it's a really pale pink. This is plasma, And this is red blood cells. Now there's a small layer in the middle, which I can't even really draw. We'll draw it in dark green but it's a white layer, and this is called the Buffy layer. I don't think it has anything to do with vampires. That reference may be too old for y'all. But it's called the Buffy layer, and this is white blood cells. This makes up about 1%. So you've got about 50% plasma, 50% red blood cells, and 1% white blood cells. I know that equals 101%, so these are a little less. This could even be a little bit more. Now, I mentioned a moment ago the different types of chains, and depending on the types of chains, oxygen can have a higher or lower affinity for the hemoglobin molecule. And about 95 or more percent of hemoglobin found in adults is composed of 2 alpha plus 2 beta, like is shown here. Where you run into differences in this is in fetuses. Now this is called hemoglobin A. A little bit of your hemoglobin is hemoglobin A2, which contains 2 alphas and 2 deltas. But both of these together share very similar affinities for oxygen. They're P50, which is the concentration at which 50% of hemoglobin is bound. So the concentration of oxygen at which 50% of these molecules will be bound is 26.8 partial pressure of oxygen. And we'll talk about partial pressure a lot more, the respiratory system, but for now just know that it is a measure of oxygen concentration in a solution. Another type of hemoglobin, which is HbF, which is fetal hemoglobin. This is found in fetuses. It is made up of 2 alpha subunits and 2 gamma subunits. The P50 of fetal hemoglobin is about 19 pp of oxygen, meaning that at a lower concentration of oxygen, fetal hemoglobin is 50% saturated with oxygen. What this tells us is that fetal hemoglobin, or HbF, has a higher affinity for oxygen, because at a lower concentration or lower levels of oxygen, it's going to be halfway saturated. And we talk about halfway saturated because it's really hard to get the full saturation. And I'll show you a graph in just a moment that illustrates this. So here we have a graph illustrating the concept that I was talking about with fetal hemoglobin versus maternal or adult hemoglobin. And you can see here, this is the saturation from 0 up to 100, or it's the fractional saturation instead of percent. You can just multiply by 100 to get the percentage. And you can see fetal hemoglobin right here in pink increases at a much more rapid rate to reach saturation well before maternal hemoglobin. And this is extremely important because the fetus isn't breathing. right? There's no oxygen going into the fetus's lungs being inside the uterus. So what has to happen is the maternal blood has to go in and mix with the fetal blood. When it does this, because the fetal hemoglobin is made up of 2 alphas and 2 gammas instead of 2 alphas and 2 betas, it has a much higher affinity for oxygen and will steal the oxygen away from the mother's blood very readily. The oxygen is just going to jump right from the maternal blood over to the fetal blood. So in this way, the fetus and the mother's body ensures that the fetus can get the oxygen that it needs to survive. Now, in different organs of the body, hemoglobin can be derived or broken down. In the liver, iron is stored as ferritin. And ferritin is a protein with an iron core, and this is used to synthesize hemoglobin. And in the spleen, which is primarily responsible for breaking down and recycling red blood cells, hemoglobin is converted to bilirubin. And bilirubin is excreted either by the kidneys or the intestines. So a very devastating disease that results from deformations in the hemoglobin protein is called sickle cell anemia. And sickle cell anemia, as you can see here, is caused by a single point mutation where you change an A in the DNA to a T, which results in glutamate getting substituted by valine. And if we look at the structure of glutamate right here, which is negative, and valine, which is hydrophobic because you have all these carbons here instead of all these oxygens here, this causes the protein to be much more hydrophobic in one place. And what happens, because the red blood cells are filled with only hemoglobin, it distorts their shape, and through the small capillaries, which are so tiny, really only one red blood cell can fit through at a time in those little stacks called rouleaux that we learn about very briefly in anatomy. They can't fit through the capillaries. And what happens is you end up with small blood clots and traffic jams of red blood cells all through the capillaries, and this can cause intense pain and actually be deadly. Now, a few other disorders with blood, primarily focusing on red blood cells. One called polycythemia. This is the presence of too many red blood cells. This can be caused by genetics, can also be caused by dehydration. When it's caused by dehydration, it's called relative. For obvious reasons, you really don't have too many red blood cells. You have too little blood plasma. Also, this can be synthetic, or you could actually say iatrogenic, which means caused by therapy, we'll say, caused by medicine. If you take your blood out, remove some blood, recover, and then add back in the red blood cells that you took out, that's going to cause polycythemia. This is what Lance was doing. In addition to taking synthetic EPO or epoetin to cause his body to form new red blood cells synthetically. Now, platelets we talk about in terms of clotting, which is what they do. They're a primary stimulator of blood clotting. We call this hemostasis. Platelets can release serotonin, which makes sense because this causes vasoconstriction. If you have a place where you have hemorrhage going on, which is abnormally high blood flow, you want to tighten down those blood vessels to reduce blood flow to that area. Also, intact endothelial or intima cells make a molecule called prostacyclin. And this prevents platelets adhering to blood vessels. So it prevents those platelets from adhering to blood vessels. Another thing platelets do, we'll change color just so it doesn't get too confusing here. Platelets also reduce inflammation, and this can also help with hemostasis. But one thing that you may have heard of people using platelets for is platelet therapy. Tiger Woods used this, I believe, on his knee, maybe his back. But if you inject platelet-rich plasma or platelet-rich serum into joints that are highly inflamed, this can reduce the inflammation in that area, which can really help with healing. So as platelets, I'm going to erase a few things because I need a little bit more room. Well, I'll just go to a new page. As platelets form a clot, they promote the release of thrombin, which is an enzyme that converts fibrinogen to fibrin. And fibrin is a protein that causes a clot in the blood. Now, a couple of drugs you may have heard of are heparin. And what heparin does is it inactivates thrombin, preventing that clot from forming. And another drug, warfarin, binds to and blocks something called vitamin K. Now, vitamin K is an important cofactor in coagulation that allows these enzymes to work. Involved in clotting. And finally, a drug that I've mentioned before, which is called EDTA. And EDTA is a calcium chelator, which means it sucks up and inactivates all the calcium in a solution. So if you put EDTA into blood, and this is a chemical that we typically use in samples of blood, calcium is involved in clotting. It needs to be present for clotting to work. So if you suck up all of the calcium, EDTA is going to block clotting as well. So these are three different drugs, EDTA, warfarin, and heparin, all of which block clotting pretty severely. Warfarin is rat poison, one of the worst ways to die. Don't put out warfarin for rats. Don't eat it yourself either. Heparin can be used as a therapy. It's also used in many blood samples. And EDTA is primarily used in blood samples and all three of these prevent clotting. Another thing that prevents clotting is aspirin. Now, aspirin blocks an enzyme called cyclooxygenase. Cyclooxygenase promotes platelet plug formation. So by blocking this, aspirin works as a very weak blood thinner. Individuals with some very minor heart disease, they'll say take a low dose of aspirin every day. This is to block the enzyme cyclooxygenase, which can then lead to a decrease in blood clots, which can thin the blood, which can help out with some minor forms of heart disease. Okay, that's it for blood. Thursday we will start to discuss respiration. And that'll be the last thing we go through because we only have 3 days left. I will probably post a lecture tomorrow on the respiratory system just to introduce us to some things, but then Wednesday really talk about the details of the respiratory system.